Sperm and egg, the fusion. The fusion of sperm and egg, known as fertilization, is a remarkable and intricate biological process that marks the beginning of a new organism's development. In humans, this crucial event typically occurs in the fallopian tube. Here's a breakdown of the key steps involved in this fascinating fusion. Sperm transport and capacitation. Millions of sperm are ejaculated into the female reproductive tract. They must navigate through the vagina and cervix, which present various challenges, including an acidic environment. Only a small fraction, around 200, reach the fallopian tubes. During their journey, sperm undergo a process called capacitation, which is a series of physiological and biochemical changes that make them capable of fertilizing an egg. This involves increased motility, hyperactivation, and changes in their membrane that prepare them for the next steps. Egg transport and ovulation.
Approximately once a month, an egg secondary oocyte is released from an ovary during ovulation. Finger-like projections called fimbria at the end of the fallopian tube sweep over the ovary and guide the egg into the tube. The egg travels through the fallopian tube, where it remains viable for fertilization for about 12 to 24 hours.
Sperm-egg encounter and adhesion. Sperm are attracted to the egg by chemical signals, chemotaxis, released by the cumulus cells surrounding the egg. The sperm first encounter the corona radiata, a layer of follicle cells surrounding the egg. They then reach the zona pellucida, a thick, glycoprotein-rich outer layer of the egg. Specific receptors on the sperm head, e.g., izumo-1, bind to glycoproteins, e.g., ZP2 and ZP3, on the zona pellucida, ensuring species-specific recognition. Acrosome reaction and zona pellucida penetration. The binding of the sperm to the zona pellucida triggers the acrosome reaction. The acrosome, a cap-like structure on the sperm head, releases hydrolytic enzymes, sperm lysins, including hyaluronidase and acrosin. These enzymes help the sperm to digest a path through the zona pellucida. Sperm-egg membrane fusion. Once a single sperm successfully penetrates the zona pellucida and reaches the egg's plasma membrane, ulema, their membranes fuse. This fusion typically begins in the equatorial region of the sperm head. Proteins like izumo-1 on the sperm and Juno on the egg are crucial for this membrane adhesion and fusion. The exact mechanism of membrane fusion is still being researched, but it involves the mixing of lipid bilayers. Prevention of polyspermy, cortical reaction.
To ensure that only one sperm fertilizes the egg rapidly undergoes mechanisms to block additional sperm entry. This is known as polyspermy block. Upon sperm entry, a wave of calcium ions is released within the egg, triggering the cortical reaction. Cortical granules, located just beneath the egg's plasma membrane, release their contents into the space between the egg membrane and the zona pellucida. These enzymes modify the zona pellucida, causing it to harden and become impermeable to other sperm. The zona reaction. Egg activation and pronucleus formation. The entry of the sperm activates the egg to complete its second meiotic division, which was previously arrested. This results in the formation of a haploid ovum and a second polar body. The sperm nucleus decondenses and swells to form the male pronucleus. The egg nucleus also forms the female pronucleus. Both pronuclei are haploid, each containing 23 chromosomes.
Syngamy and zygote formation. The male and female pronuclei migrate towards each other in the center of the egg. Their nuclear envelopes break down, and the chromosomes from both pronuclei intermix and align. This fusion of genetic material is called syngamy or karyogamy. The result is a single diploid cell with 46 chromosomes, 23 pairs, known as a zygote. The zygote contains all the genetic information for a new individual.
The zygote then begins a series of rapid mitotic divisions, cleavage as it travels down the fallopian tube towards the uterus, eventually forming a multicellular structure called a blastocyst, which will implant in the uterine lining to initiate pregnancy.